Your question, please.
I want to ask if you consider something like metabolic rate of cities. If there is some break point between small towns and big cities, if it's something that is possible to come.
Yeah, that's a great question, and I've thought about that. It was just a workshop actually on scaling in cities, so and I was one of the few biologists there. So I was looking at data and I didn't see any clear indication of like a strong shift in the slope, but what I did see was that a change in the variance. So in the smaller cities, for example, carbon dioxide emissions varies a lot more in smaller cities, whereas in larger cities it fits much more tightly along the scaling line. And the scaling relationship is, at least in these US cities of this paper, was roughly like 0.9, so it was roughly linear, maybe slightly sublinear. But I think that the change in the variance is also maybe informative, but that might suggest that the drivers of CO2 emissions are different. So at smaller cities, it's more just, people are just, they behave how they're going to behave. They drive their car or whatever, and so there's no fundamental constraint like at the supply level in terms of CO2 emissions until you get to really large cities where then not everyone can drive, and there's more public transport and things like that, and the scaling, the CO2 emissions ends up being much more tightly falling along the scaling line. Yeah. Yes?
You showed that heterotrophic unicellular eukaryotes scale have linear scaling.
Yeah.
Metabolic scaling. Your data imply that phototrophic unicellular eukaryotes should have sublinear scaling, given the fact that there was a lower slope of that number of mitochondria versus in phototrophs.
Yeah.
But you did not show any data.
Yeah, that's right. Yeah.
But if that is the case, should the phototrophic unicellular eukaryotes have higher rate of employment, the other mechanisms of how attain, let's say, linear scaling, such as surface fractalization or how we call that, geometry dissimilitude? Yeah. Then the heterotrophs. And are there any data that could illustrate that?
Oh, yeah, that's a great question. First of all, there are data suggesting sublinear scaling in chloroplasts, in phytoplankton, both in terms of like growth rate as well as respiration or photosynthesis rate. So I just didn't show the data because I was trying to keep the amount of plots to a minimum. In terms of like strategies or adaptations to that sublinear scaling, there are a few things that those cells do. The chloroplasts tend to be located closer to the cell membrane rather than the interior of the cells, especially in the larger phytoplankton. What else? The really large ones will have cytoplasmic streaming sometimes. So it's essentially they're actually actively pushing, moving liquid throughout the cell. So it's like a very primitive vascular transportation system in that sense that you don't ever see in really small cells. Yeah, and I think there's opportunities for thinking more about other kinds of strategies. Certainly a lot of the really big phytoplankton have, like right here, these are radiolarians, which you can, so these are actually unicellular eukaryotes that have very extreme levels of geometric dissimilitude. So they're forming these star-like shapes. And so there's other examples like that in diatoms and dinoflagellates.
Next question, please. Meanwhile, maybe I can have one.
Yes, please.
So my question is, you showed the proportional relationship between number of mitochondria and cell volume, in multicellular organisms. So my question is, in which cells is it Is it one kind of cells because we have a lot of cell types?
Oh, yeah. Great question. Yeah, I should have.
Blood cells, or I don't know what that is.
Yeah, that's a great question. So it was a variety of cell types, but it was also constrained by the data. So there's more studies on certain kinds of organs, for example. But so liver cells, kidney cells, different muscle cells, lymphocytes. This is in animals, and then plants, different parts of the leaf. So there's different cells, depending on where you are in the leaf. And so I just tried to get a great variety of different cells to try and understand then the degree to which a cell being in this ecosystem of an animal or plant, how it's affected by that constraint.
So in the figure there were several points from human beings, say?
Yeah, so in the figure for the plants and animals, each point is the average for a cell type for a species. So then there's variation both across species within a cell type as well as variation across cell types within species.
So, other question, please? Looks like having something in mind.
I have such a very unpolite question.
Oh, no, that's good.
Where are dashed line with 95 confidence intervals? Because the slope can be anywhere.
Yeah. So I guess in the published stuff that I showed, there were confidence intervals on there. I didn't always show the lines itself for the confidence intervals, because then if you have showing a lot of different groups, it leads to plots being way too busy. So it just showed and reported instead of the confidence intervals within the figure. But then when I was then talking about these hypotheses and theories, and that's using other people's data, and so that's more, I was not trying to imply that I had done a rigorous statistical test yet of the ideas. Yeah.
I think we can discuss about this different shape and this different transition between different slopes.
Yeah.
We can discuss this, but all these slopes can be within this confidence, I suppose.
Right, yeah. Well, when it's my data, what I was doing is using these general linear models. So it's sort of like an ANOVA, right? And that tests whether or not there's a difference in slopes between groups. So it takes into consideration the confidence intervals. Yeah. And then it provides a p-value, right, and that kind of thing. So. Yes.
If I understood it correctly, you said that in prokaryotes, larger genomes cause larger metabolic rate. I wonder if you have any comment why in multicellular eukaryotes there is some researchers believe negative correlation between genome size.
They believe what?
They believe that there is a negative correlation.
A negative correlation. Oh. Metabolic rate of what? The whole body, whole organism metabolic rate, they think is so. But they're looking at bodies of the same size. Then are they controlling for variation in body size, or because otherwise it'd be okay? So if you just plot genome size in birds, there's this roughly negative correlation. Even though a tiny bird is obviously going to have a lower metabolic rate probably than a big bird, even though it might have a bigger genome size, right? You're saying so after you account for body size effects. It could just be, so a lot of the processes involved in maintaining a genome are metabolically expensive, and so you have a larger genome, there's a lot more DNA that has to be continually repaired, so that's going to require a lot of energy. The replication of the genomes through mitosis. If you have a bigger genome, it requires more energy to replicate, and so the cells also sometimes have to be bigger with bigger genomes, and that could also maybe contribute then to the metabolic rates.
Okay, so I'd like to ask whether you studied if there is some difference difference in the variation of the measured values of the metabolic activity between prokaryotes and eukaryotes.
Oh, and the variation? Yeah.
The variation of the
Like between physiological states? Yeah, that's interesting. I haven't done a test on that, but that would be interesting to do. Yeah, to see whether or not the variation like changes or not across these groups. Yeah, it's a good idea.
Okay.
Yeah, if you look at the lines, it doesn't look like it changes very much, certainly, because they were pretty close, you know, on this broad scale. But yeah, that would be interesting to do. Yeah. Yeah, and I think, yeah, that would be what I would expect as well, just because prokaryotes use such a greater diversity of different kinds of substrates, and so sometimes they're trying to digest things that, you know, no animal can digest, right? And so that leads to different energy yields, and that could then just lead to greater variation in metabolic rates.
I have one related question or comment. So to me it seems that like proportionality between metabolic rate and cell size in protists, it's like nature. It's like neutral expectation.
Yeah.
And negative relationship quite slow. It is, yeah, we all know it. So sometimes it could be explained by increasing number of inert metabolic tissue, like bigger skeleton, elephant, relatively to its body volume than in mouse, say. However, the really tricky is the positive allometry of metabolic rate in prokaryotes.
Yeah.
It is something really strange, and I think couldn't it be related to number of metabolic pathways, because it's really difficult to measure metabolic rate when these bacteria utilize ethanol as substrate and these utilize like glucose or something. So how these differences in different substrates of energy, they are heterotrophs, but did they use only a single metabolic pathway to get energy, or were they fed by different lactose and another substrate?
No, they do have to be fed different substrates in certain cases, because each one might be evolved to different food, just like you wouldn't try and feed a carnivore grass and so the food has to be adjusted in that sense. But these are all heterotrophs, so they're all fed organic carbon. But then certainly within that, there's such a, even for a given substrate, then there's different ways that substrate can be broken down. So there's different metabolic pathways to break that. And some ways might have a higher energy yield more ATP per unit substrate, but some might have a higher rate of substrate use because of
the metabolic rated consumption of oxygen.
Yeah.
But it can be.
These are all aerobic. Yeah.
Among different pathways, how much energy you need from per single oxygen.
Yeah.
Okay. Isn't there any trend between small cells and big cells? For example, big cells use ethanol and small.
Oh, there is. Yeah, no, definitely. I showed some data showing that the number of metabolic pathways increases with cell size in prokaryotes. So the really large cells have the largest genomes in prokaryotes, and they tend to be evolved to environments that have a great diversity of different substrates, like soils. Whereas the really small prokaryotes tend to either be oligotrophic organisms, like in the ocean, phytoplankton, tiny little cyanobacteria, or parasitic prokaryotes that have really reduced genomes, because actually some of the metabolic reactions that would be required to sustain themselves are being done by the host organism, not by themselves. So they're able to have these reduced genomes and metabolic networks.
Yeah, I see. And related issue is that in the small genomes in prokaryotes, so probably they are so reduced that all gene has to be transcribed all the time. However, if you have big bacteria, they have some redundant genes because they do not utilize now lactose as everybody knows. So some genes are switched off, and it should make some difference, isn't it?
Yeah. I think that's. I think that could also certainly help, having a greater number of copies of different genes, so that that's not a rate-limiting step in metabolism and the process of transferring, information to enzymes. If you have a greater number of genes, of copies of those genes, then you can transcribe and translate much more quickly. So it's hard to
substrated, but mycoplasma only single ones.
Yeah, so that could play a role. Of course, the question is whether it's just responding to some other driver, or whether it's actually constraining the metabolic scaling. And it'd be interesting to try and tease that apart with maybe experiments or more detailed kind of modeling of a cell and looking at all these different factors that might constrain metabolism.
Okay. So then I would say that the data in bacteria are more messy. In fact, the variance can be higher because there are many groups that are different, have different metabolism, and what we are comparing.
I've gone back and forth on that, because in a way that's true, but if you think of single-celled eukaryotes and even plants and animals, well, not plants, but animals, so there's a lot of messiness there in terms of what they're fed, whether you're a tapeworm versus an elephant, that's also quite different, right? Whether it's a parasitic eukaryote, like malaria or something, versus a large eukaryote that has a lot of mitochondria that's in the aquatic system or something like that. So there can be a variety of different of niches and
metabolic pathways in bacteria is much higher.
Yes, definitely. Yeah, and that's probably part of why there's this difference in scaling, I think. Yeah.
Okay, so next comment, please. I'm sorry that I occupied your time for such a long time. So, not about protists even. Okay, so thank you very much. Thank you once more.
Thank you.
And come next week.